Good evening, everyone. My name is Gene Robinson. I'm the director of the Carl Woese Institute for Genomic Biology, and it's my pleasure to welcome you tonight. I want to welcome members of the Champaign-Urbana community, members of the campus community. I want to welcome our distinguished guests, several who are welcoming back home: Harris Lewin, the founding director of the Institute, Larry Smar, the founding director of NCSA, and all our distinguished speakers for the weekend. So we're here to kick off an amazing event, a symposium to honor one of the most illustrious faculty members at the University of Illinois, and someone who was one of the most impactful and influential scientists of the 20th century, Carl Woese. You'll be hearing a little bit about Carl from Nigel Goldenfeld in a moment. We were really proud to rename the institute the Woese Institute for Genomic Biology. We will still be known as the IGB, in case you're worried. We did decide, though, that if we ever go into the country music radio stations, we're going to be called the WIGB. But for now, we're going to stick to science, and so we are known as the IGB. We have a really amazing speaker tonight, Penny Chisholm. We're delighted to welcome you, Penny, and thank you very much for coming. And now it's my pleasure to introduce Nigel Goldenfeld, a professor, a Swanlund Chair, which is the highest honor that can be bestowed upon a faculty member here at the university, a member of the National Academy of Sciences, a distinguished professor of physics, and one of the theme leaders at the IGB, the theme called biocomplexity. Nigel is a longtime collaborator of Carl, and is very influential in moving the entire discipline of biology towards a more quantitative approach. So Nigel's going to come up and tell you a little bit about Carl, introduce our speaker. I just want to mention two things quickly. There's a reception after the lecture tonight. You're all welcome to that. And then the symposium kicks off tomorrow morning at 8:45 in the same room. You're welcome to come and go as you please. Listen to as many of the amazing lectures that will be held all day tomorrow and Sunday morning. So sit back and enjoy a really great lecture tonight. Nigel?
Thank you, Gene. So as you've heard, this weekend we are hosting a major scientific symposium to celebrate the rededication of our institute, henceforth to be known as the Carl R. Woese Institute for Genomic Biology. Many of you will know that Carl Woese was one of the most distinguished scientists to work at our university, famous for groundbreaking discoveries in evolution and microbiology that overturned our previous understanding of such basic concepts as species. Now, every once in a while, you'll read in the newspapers that a new species has been discovered. Maybe someone finds a new type of butterfly living in a forest, for example, or a new species of lizard. Carl discovered something unimaginably greater, a whole new category of life that existed alongside the two types of organisms with which we were previously familiar: bacteria, which you generally can't see, and eukaryotes, such as you and me, which you can. Today, we call his discovery the third domain of life, and the category of life that he discovered is generally known as archaea. However, the real importance of his discovery was how he made it and why. Carl was a physicist by training, and he wanted to map out the evolutionary history of all life on Earth. He was a humble man personally, but he had lofty goals, and he was not afraid to embark on something that was unprecedented and very risky. To make his map of life, he decided to use the chemical sequence of a fundamental component of every living cell, something that all life must possess. His emphasis on molecular sequences was in its way the progenitor of today's genomic revolution, and led to a transformation in the way that biology was done henceforth. None of this was an accident. Carl understood decades ago the way in which all this would play out. And while his contemporaries were absorbed with the structure of biological molecules, Carl was busy laying the foundations for a new biology. For this reason, our symposium is called Looking in the Right Direction, Carl Woese and the New Biology. We are starting our symposium with this evening's public lecture. The impact of Carl's work and the work ongoing at our institute resonates through to everyday life and is already beginning to have a transformative influence on healthcare, ecology, and perhaps even understanding the full implications of global climate change. And no one is better qualified or more appropriate to lead us in the right direction than tonight's speaker, Professor Penny Chisholm, the Lee and Geraldine Professor of Environmental Studies at MIT. Like Carl, Penny made a fundamental and surprising discovery about microorganisms, in her case, a little microbe found in the sea. And like Carl, she has mined this discovery in every way imaginable, transforming our understanding of microbiology, the relationships between microbes and the viruses that prey on them, and the way in which astronomically, or rather cosmologically, large numbers of cells can have a major effect on the chemical composition of not only the oceans, but also the very air that you are breathing. Penny's work has been at the forefront of efforts to collect massive amounts of molecular and genomic sequence data that can be cleverly interpreted to explore in great detail how the Earth works. And she is a pioneer of the burgeoning field known as metagenomics. Penny's work not only informs the current public debate about global climate change, but is a critical ingredient of the debate about how one might try and mitigate its effects by intervention or not, something about which she has been very vocal. Penny is not only a fantastic scientist, but she is also an author, not just of scientific papers, but of 2 award-winning books for children that explain how sunlight drives the oceans and the plants. Penny has received many major honors in her career, and I don't have time to list them all. I will, however, mention that, like Carl, she is a member of the US National Academy of Sciences, and, like Carl, she has received the National Medal of Science, in her case from President Barack Obama. This is the highest recognition that is given to the nation's scientists and is a fitting tribute to her scientific contributions. We are thrilled, Penny, that you are able to join us and our other distinguished guests and speakers at this weekend symposium, and we're equally thrilled that you are here to talk tonight. So please join me in welcoming Penny Chisholm.
Well, now I'm really intimidated after that introduction. I tend to walk around, so I'm going to tuck this away. Well, it's really an honor and a pleasure to be here tonight to celebrate the life and work of Carl Woese. I've never actually met Carl, but in the decade before he died, we had a fairly regular email correspondence. And it started out just as a casual, something about the National Academy of Sciences. He was championing somebody, he wanted to be elected. And it slowly morphed into, I better get away from that. It slowly morphed into talking about science, and I realized that he'd actually, it's bad? Okay. He's working on it? Well, I'll go. Does this work? Here we go. Okay. Anyway, in the emails exchanges, I realized that he had actually read the papers that came out of my lab, which was totally thrilling to me. It's just weird to realize that somebody actually reads them and even likes them. So then we started talking about science, and I came to realize that his view of life on the planet was very similar to the way I thought about it. Now, he is coming from hardcore physics, and mine was coming from sort of intuition from studying this one single microbe for 25 years. So that was just a very inspirational exchange of emails. And when I started preparing this talk, I went through 10 years of emails from Carl, and it was really, he came alive again, and I pulled out some quotes, which I will scatter through my lecture. And I hope that will help bring him into the room. In thinking about shaping this lecture, it's really hard to figure out who's out there, or who is going to be out there. I think everything from people who are experts in better experts in things that I'm going to be talking about, to people who maybe don't even have a science background. So it's impossible to hit all of those things. I hope I find a happy medium. But I decided to tell a story. I think of science as a story. And everybody likes stories, so that's what I'm going to do tonight, and that's going to be the story of Prochlorococcus, which is this tiny microbe. It's the most abundant photosynthetic cell on the planet, and how we discovered it, and all of the things that it's taught us in studying it over the years. And it's going to be my personal journey. I'm not doing a review of everything that's known about Prochlorococcus. But I hope you like it. And one of the quotes I dug out from my emails from Carl is that when he once described the Prochlorococcus story as a fresh walk in the garden of evolution. So I hold on to that. So here we go. First, the fellow travelers. These are all of the people who have worked on this organism in my lab over the years. And to steal a line from Simon Levin, when I say I mean we, and when I say we, I mean they. They're the ones who have really done all the things that Nigel just gave me credit for. And they come from many different disciplines, and that the lab has, over the years, been very interdisciplinary, and that I think is what's contributed to some of our successes. And also I need to recognize the benefactors of all of this work. I do this in the beginning of the lecture because I will run out of time at the end, and I want to make sure to acknowledge all of the people that have funded this work. And then Carl acknowledged as a guide, and this is one of my favorite quotes from him. It's our task now is to resynthesize biology, put the organism back into its environment, connect it again to its evolutionary past, and feel that complex flow that is organism, evolution, and environment united. And that's the framework in which we've been studying Prochlorococcus all of these years. So the itinerary of today's lecture, first I'll talk about the big picture, life on Earth, and then I'll describe the discovery of the serendipitous discovery of Prochlorococcus and what we've learned from this microbe, and then talk a little bit about reframing the life sciences. So whatever talk I'm giving, I start with this slide because there's nothing more important on Earth, as far as I'm concerned, than photosynthesis. The editor of our children's books disagreed with that, but I fought hard. Because without plants and photosynthesis, nothing else, well, there might be a few microbes eking out a living, but the majority of life relies on this sunlight-driven drawing of carbon dioxide out of the atmosphere to make plant biomass, which feeds the rest of living creatures. And all of us, plants and animals and microbes, then burn that biomass, sugar, to stay alive and regenerate that carbon dioxide and water. And oxygen is evolved as a byproduct. And then there are these microbes that recycle all of the elements that are necessary for these processes to go on. And this is so extraordinarily important to realize, which is what drove me to write these children's books on photosynthesis, is that people don't realize. In fact, they did a study of Harvard and MIT graduates and said they had a seed and a log. And they said, here's a seed, here's a log. Where does the weight of the log come from? None of them answered from the air, because it's just too hard to think about that. That weight actually came from the air. So photosynthesis, very important. And half the photosynthesis on Earth is done by phytoplankton. These microscopic plants that live in wherever there's water or ice, seawater, freshwater, phytoplankton are there. And they fix 35 to 50 gigatons per year of carbon from the atmosphere. And for lay audiences, I put this in Volkswagen units. So that's the weight of 50 billion Volkswagens. Is what these microscopic plants draw from the atmosphere and turn into biomass. Even though they weigh globally only a thousandth of all of the land plants. They're busy doing photosynthesis and feeding the food webs of the sea. And of course, the fossil fuel is fossil phytoplankton or algae, and what we're doing now is accelerating this respiration term by burning this fossil fuel and increasing it in the atmosphere at a very steady rate. But I'm not going to cover that tonight. I'm going to go on to the journey of Prochlorococcus. So before 1979, our image of phytoplankton in the oceans was something like this. There were some big colonial ones. This is colonial cyanobacterium. You can actually see it with the naked eye. And then a lot of different cells, say between 10 and 50 microns in diameter. If you're not used to thinking in terms of microns, well, here's the discovery in 1979 was that there are tiny, tiny bacteria, cyanobacteria, blue-green algae in the oceans that are only 1 micron in diameter, really small and very abundant. So 100 of these would line up the width of a human hair. And that was a big paradigm shift. That was the big discovery of these things that are called picoplankton. But John Waterbury and Woods Hole and colleagues discovered them. And at the same time, about that time, Rob Olson was a postdoc in my lab, and we were using this instrument called a flow cytometer, where you could inject samples of cells into a fluid, sheath fluid, flowing fluid. They would be focused and go through a laser, and they would scatter light and fluoresce, either if you added a stain or cells that have photosynthetic pigments autofluoresce. And so we thought, well, this would be really cool to put on a boat, since the seawater is just filled with these fluorescing phytoplankton, and just see what we could see. These instruments were developed for biomedical research, cell biology in hospitals. So putting it on a boat was not a small feat, especially when you have a laser focused on a tiny capillary tube. And I credit Rob Olson with that adventure, because there's no way I would have done that. I could encourage him to do it, but it was scary. So, because this is what it's like at sea sometimes on a boat, and there's Rob with the flow cytometer scratching his head because it wasn't working. But he managed to make it actually function. We bought it with a money-back guarantee from the company that if it didn't work on a ship, they would take it back. And we started studying these little, tiny little cells called Synechococcus that John Waterbury had discovered. And so here's a typical flow cytometric signature. This is forward scatter, that's a measure of size. And this is fluorescence. And this is a population of Synechococcus fluorescing orange, because they have a special pigment that fluoresces orange. And we were studying them, and we noticed something strange. And I get the credit for discovering this, but I credit Rob Olson for discovering them. And if I had time to tell you the whole story, they were discovered. This was the third time they were discovered. So they were actually discovered before, but people didn't realize they discovered them. So, but that would take me another 15 minutes. So this is electronic noise, and emerging from that noise were these tiny cells, smaller than these 1 micron ones, that fluoresced red instead of orange. And we were pretty convinced that was something alive, because it got more abundant with depth. And we called them unidentified picoplankton. That's the first name we attached to them, because we had no idea what we were doing. Then we started to call them little greens, because we suspected they were maybe green algae, because they had this fluorescence that was characteristic of that. And Rob managed to take a picture of them with the microscope, and this is a fluorescent microscope. They're green colored, but they fluoresce red because of their photosynthetic pigments. I'm going to move this here. There. And so the next slide is that same picture, but under a light microscope. And there they are. And that tells you why nobody knew they were there before, because they're very hard to visualize under the microscope. So this is the 1988, it's a birth announcement. We published a paper on this, which we called a prochlorophyte. And they're less than a micron. They're about 600 nanometers. And a physicist once pointed out to me giving this talk that they're the same size as the wavelength of light that they are absorbing. And I've always thought that was kind of cool, so I always show that. Not that I just have this image of this cell with this wave of, I don't know, whatever. But but they contain, we were able to determine from the samples in the sea that they contain chlorophyll B, which is the accessory pigment that higher plant chloroplasts have. And these chloroplasts are known to be evolutionarily derived from cyanobacteria, which is the group that Prochlorococcus belongs to. And over evolution, they originated another cell, engulfed them, and they became symbionts, and then they became these chloroplasts. Anyway, so we got excited because we thought maybe, that we were seeing the close relative of chloroplasts. And so, because here's a picture of Prochlorococcus culture more recently taken by MBL, and here's a picture of higher plant chloroplasts. They look similar. And here's where, thanks to molecular evolution, Carl Woese's major breakthrough in the taxonomy of life, we were able to sequence those ribosomal RNA with the help of Mitch Sogan, because I'm trained as an ecologist, so this was my adventure into molecular biology. Luckily, I had a graduate student from the biology department at MIT who had studied molecular biology, and so she was very confident, and she knew what she was doing, but I was supervising her, and I didn't know what she was doing. So that was the first time that had happened. That's happened with every student since then. But because I have jumped right in doing everything that I'm not trained in. But Mitch held my hand through that, and I realized the power of this to help us identify the origin of these microbes, which we now call Prochlorococcus. And so this is the tree that Ina Urbach was a graduate student, and Brian Palaniuk also sequenced another gene to make a tree. But anyway, long story short, they're not anything special relative to chloroplasts. They're just a close relative of Synechococcus that evolved chlorophyll B independently. So that turned out to be not earth-shattering or exciting. So we had to find something else to get excited about. So we just decided we were going to just study Prochlorococcus. And here's my first Carl quote. Beware of Darwinian snakes and phylogenetic trees, which is something he sent me in an email once, and I've pondered ever since, but I can't look at a tree without hearing that in my head. All right, so fast forward, now it's 2013, and this is a paper by Adam Martini's group where they took all of the census data on Prochlorococcus over the global oceans. These are all the cruises that have looked at it. And the estimate is that there are 3 billion Prochlorococcus, and that's 10 to the 27th, in the global oceans. And I like to express this unit per planet, because we can do that, because we know where they are. And we have a pretty good idea how many there are. Up to 700 million cells per liter, and they're globally estimated to fix 5 gigatons of carbon per year. That's 5 billion Volkswagen units. These tiny little cells are sucking out of the atmosphere of CO2. So the world of Prochlorococcus, we spend a lot of time imagining what it's like to be a Prochlorococcus cell. And they're very simple. They're tiny. They grow by dividing in half. But they get eaten just about as fast as they grow. So their doubling time of the population is once every 1 to 3 days. But their population numbers, I'll show you in a minute, stay fairly stable. So it's a very rapid turnover. They also get infected by viruses. That's another source of mortality for them, and I'll talk about that a little bit later. And their environment is extraordinarily dilute. There are what we call picomolar concentrations of metals in the open ocean, so dilute that we can think of these cells almost as bumping into atoms one at a time when it comes to trace metals. I mean, that's how dilute it is. So, as I said, they're turning over very rapidly, but they're very stable over decades from ocean to ocean. So we started in 1990, 1994, that's these gray numbers. This is a time series of Prochlorococcus abundance off Bermuda and off Hawaii. And then we started up again in 2004 for another 5 years for a different reason. And you can see that they're just out there doing their thing day in and day out, dividing, being eaten, dividing, being eaten. And it's just amazing. And when you think of that they're embedded in this complex microbial food web, details aren't important here, but the slide I showed you before was really oversimplified. I mean, there's a lot going on out there, and yet it's beautifully stable. So that's what we spend a lot of time thinking about. How does that work? Okay, in 1990, after many years of trying, this is Brian Palaniuk, who was a graduate student in a colleague's lab, Francois Morel's lab at MIT, succeeded in actually getting these cells into culture. So everything that we learned up till then, we were studying them in the seawater. We'd collect seawater and we'd do experiments, we'd perturb them, we'd see how they reacted, and we'd try to extract things from them and figure out what was going on. So finally, we got them into culture. And that's when I decided this is what I was going to do for the rest of my career, was study them. Because I had been frustrated with other species of phytoplankton, trying to figure out, you could study them in the lab, but you couldn't go out the ocean and find them. Or there are things you could study in the ocean, but you couldn't bring it into the lab. So this was one that we could now study as a simple microbe. It was really widespread. You could find it where it lived. It would always be there in the numbers you wanted it to be there. It was easily studied in the sea. We could cultivate it. And most importantly, its habitat is measurable. So you could collect samples and you could study the seawater, the chemistry of the seawater and the physics where you collected them. So you could relate their properties to the environment that was shaping those properties, which is difficult to do for a lot of terrestrial organisms. Granted, you have to go out on a ship, but it's fun. Some of my former students are laughing at that. Okay, so the habitat that they live in, they live in the upper 200 meters of the oceans. The oceans go down to 4,000 or 5,000 meters in places. And it's only this thin film on the upper surface that's doing all this photosynthesis, feeding the entire ocean. And so there's this surface mixed layer of maybe 50 meters, and then there's a temperature gradient and a salinity gradient that separates that from the rest of the deep water. And things rain down from the surface, they're regenerated, so you have this gradient of nutrients with depth, and you also have this light gradient. Our first isolate was from the deep water. The first one we got into culture, its name was SS-120, Sargasso Sea 120 meters. And then a colleague isolated one from the surface in the Mediterranean Sea. So that was the second strain. So we started studying them in cultures and how how fast they would grow as a function of light intensity and sort of basic physiology that you do on cells. And the first thing we noticed was that the cell that was isolated from the deep water, if this is growth rate, light intensity, had an optimal light intensity for growth that was much lower than the one that was isolated from the surface water. In fact, these light intensities would kill the deep one, and this one would grow at light intensities that this one could not live at. And these would be considered the same species in traditional microbial speak. So they're dramatically different. And using molecular probes, again, inspired by the ribosomal RNA sequencing that Carl Woese brought to us, we could see that these two ecotypes, as we called them, were distributed dramatically differently with depth. The high light adapted ones were in the surface, the low light adapted ones were only dominant in the deeper water. Well, we started isolating more, and we found that they fell into these different groups, high light adapted and low light adapted. And we started looking at these closely very similar ones. And this is a transect that Zachary Johnson, who was a postdoc in the lab, did along the Atlantic, looking at, and I'll just refer to these by their colors, the yellows and the greens. Just you don't need to know their names or whatever, but and that the yellow ecotype and the green ecotype were dramatically different in their distributions along this transect. And the first thing that comes to mind is temperature. Here's the equator. So we go back to the lab and we look at the temperature optima of these two, and lo and behold, yes, the yellow one has a much higher temperature optimum, and then the green one, and this one can grow at much lower temperatures. So that's why it's the dominant one at the higher latitudes. So as we isolated more and more strains, these fell into place into these high light adapted, low light adapted group. These were much more closely related. These guys we didn't understand that much. And this is the last phylogenetic tree you're going to see in this lecture. But just to say that we were able to start to order them in terms of their evolutionary history. So we developed probes for these different clusters, these different ecotypes, and studied them off Bermuda. So just think of each one of these colors is one of these groups with a unique physiology. And notice this is a log scale. And this is several years. And you can see with the seasons off Bermuda, their abundances go up and down. And these are the low light adapted ones, and the green and the yellow, the high light adapted ones, and this blue one is actually adapted for mixing. It tolerates fluctuating light much better than the others. And you can see that these oscillations are out of phase. And in the old way of thinking, people would say, oh yeah, they're competing with each other, and this one, at this time of year, this one's winning out, so it's going up, and this one's losing, so it's going down. And we thought of competition as shaping this, and that's my excuse to bring in the next quote from Carl, which is, all the problems of modern society trace back to the Darwinian deception. And you can think about that for a while, but the point is that we don't think that these out-of-phase oscillations have anything to do with competition. They have to do with the co-evolution of the consortium of cells. And I'll show you how this is just a very, very crude cut of the diversity in this group. I'll show you the real diversity in a minute. But it's all part of one unit. And when the conditions change, there's just one that's slightly better. That one goes up a little bit. Conditions change again. And it's contributing to this stability of the population. Okay, so the classical definition of an ecological niche is, and this is what the Hutchinsonian niche, but it's called the n-dimensional hypervolume, every point on which a species can survive and reproduce. And so what I've described to you so far is the temperature and the light dimension of the niche of this group of ecotypes. And there's much more regulating the growth of these cells. So how do we get at that? Temperature and light are the obvious things to look at. Now, we wanted to understand more about the diversity. And that's when genomics entered my lab. I joke about getting sucked down the tunnel of reductionism because I was an ecologist. First we got into molecular biology, and then we had to get into genomics, but we were lucky enough to be one of the, I think we were the second microbial genome to be sequenced by the Department of Energy. And so we managed to convince them to sequence not one, but two, because they're tiny. We got a twofer. Because we knew that the comparison between this high light adapted strain and the low light adapted strain would yield so much more information than just a sequence of one, because it's the difference, it's the comparison that really tells us what's driving what's shaping these communities. So we learned quickly, even though they're the same species, they're fundamentally different. They have different numbers of genes. This low light adapted has many more genes than the high light adapted. They share about 1,350 genes between the two of them in common, and they have very different base pair composition in their DNA. They're very different. If you lined up the genomes, this is just the position of the genes against the position of the genes in these two strains. If they were identical, this would just go straight up, straight line. And you can see they're incredibly different. So that was exciting. But the interesting thing was many of these genes are genes that are of unknown function, but at least we were able to identify some that mapped on to the habitat from which we knew these cells were isolated. They had different nutrient acquisition genes, the gradient of nutrients here. The deep isolate, the low light adapted had enhanced pigment synthesis. High light adapted had more light protective proteins. We were able to suddenly start to see in the genomes the ecological features that we had been studying using physiological analyses. So then we started, sort of fast forward a little bit, then the question was, well, how diverse are they? And this is sort of my mental image, my fantasy of this is the sea filled with these billion Prochlorococcus. The question is, if you took a milliliter of seawater, and there's 10,000 cells in that milliliter, probably more than that. Anyway, lots. Are they all different? That was the question. And I don't have the answer yet, but close to it. We now have the full genome sequences of 39 strains from diverse habitats. And every time you sequence a new strain of Prochlorococcus, you get about 100 to 200 new genes that you never saw before in any other Prochlorococcus cell. So this is number of genes. This is pairwise comparison of cells. So it settles down and this is the number of genomes examined, it settles down very quickly to about 1,200 genes that they all have in common. That's the core. That's the essence of being a Prochlorococcus. But every time you sequence another one, you get more genes. And this is what's called the pan-genome. These are the flexible genes that not all the cells share. And the projection is that the Prochlorococcus collective has about 80,000 genes. That's just a theoretical projection, but that's 4 times the number of genes that we have. And so this is this is quite an organism. So we sat around in a lab meeting. I had a hard time describing this to some colleagues who work in mammalian biology, and the idea that you could have these flexible genes and these microbes that had different numbers of genes with different functions was just too hard for people to think about. So we sat around in my lab meeting, and I said, I need an analogy. And one of the students, Katia Foix-Moniz, came up with the iPhone. And it helps us understand what this means. So the iPhone, if this is a Prochlorococcus, the iPhone has 24 core apps. These are the ones that you can't delete when you... That's the essence of being an iPhone. They're hardwired into them, like the mail and the phone and the... but they have all these flexible apps that you select to custom design your iPhone. And those are analogous to these flexible genes that over evolutionary time are moving in and out and being selected for by different strains of Prochlorococcus. So here's one of these flexible genes. You know, if you're an iPhone, you know, when you want to delete an app, if you have an iPhone, you make it jiggle. It took me a long time to do this. You have to appreciate it. Then you push that X and it goes away. That gene's been deleted. Okay, so turns out there are 1.2 million apps in the iPhone pan appome. That's our new term. And that's rising, just like our estimate of the pangenome of Prochlorococcus. Those are being invented every day. But there are estimated 2 billion genes in microbes in the ocean. So the microbes still have the iPhone beat. And this is I have another quote from Carl here. We multicellulars are all islands floating in a bacterial sea. That's what I use there. Okay, so what are these flexible genes doing? And a lot of them have functions that are unknown. We don't know what they're doing, but some of them we can infer. They're involved in cell surface properties, which are involved in defense from phage. Some of them are involved in specific nutrient acquisition. Others are involved in stress responses. I didn't know how to make a stressful looking cell, so that's a stressful looking me. I think that's a... and 80% of these genes, these flexible genes, are from very distantly related organisms. So there's a lot of genes moving around out there. So the next question is, how are these genes that can some cells have and some cells don't, how are they distributed in the oceans? And that's where metagenomics enters in. And for those of you that aren't familiar with this, if you have a genome of a known organism, a cultured organism, and then if you go out in the ocean and you collect a lot of seawater and you chop up all the microbial genes into pieces that are about the size of a gene from all the different bacteria, and then by similarity, you can pull out the ones that belong to Prochlorococcus from this sea of genes that are from all the different microbes. And then you can look at those. And so the first thing you do is you can compare the relative numbers of these genes, these flexible genes, in different oceans. So this is a simplified illustration. So if this is an Atlantic sample and a Pacific sample, and here's a gene that's way overrepresented in the Atlantic. There are many, many more copies of that. And that means that more of the Prochlorococcus in the Atlantic have this gene than in the Pacific. So then you ask, well, what is that trying to tell us? What is that gene? What does it do? And this is actually the work of Maureen Coleman, who's here in the back, who was a graduate student in the lab. And she compared these families of genes, Hawaii and Bermuda, and these had a much higher frequency of phosphorus acquisition genes, the Prochlorococcus in the Atlantic station. And that made complete sense because the phosphorus concentration in the Atlantic is an order of magnitude lower than in the Pacific. So these cells have a lot to tell us about the ecology that they're experiencing. Their genomes hold the information that tell us what the selective pressures have been on those cells over millions of years. So we think about all the other genes that we don't know the functions of that have differential distributions that are really trying to tell us something, and we won't know what that is until we can understand their function. Okay. So how diverse are they? What is the finest scale of meaningful diversity in these tiny little cells? And this is what gets us into single cell genomics. So with this flow cytometer, you can sort out single cells into individual wells. You can amplify their DNA and select the cells that you want to sequence and sequence genomes. And this is the work of Nadav Kashjan, who was a postdoc in the lab some years ago, where he did this off Bermuda, studied Prochlorococcus over the season, and looked at the individual cells from a parcel of water. And I don't have time to tell you that story, but the bottom line is there's extraordinary diversity among the cells. There were thousands, we'll go back to our iPhone analogy, there were thousands, there are not only differences in their flexible genes, but also they have differences in their core genes, slight differences in their core genes, which he defined as their genomic backbone. So there are thousands of these backbone populations, and each of them with trillions of cells. So it's extremely diverse. So the way it's shaping up, the way we're looking at this is, here's the total Prochlorococcus time series off Bermuda that I showed you earlier. And I showed you this earlier, that we break that up into these 5 ecotypes, very broad ecotypes. And now with Nadav's single cell work within these ecotypes, there are really finely tuned subpopulations. So it's just extraordinary. I call it a masterpiece. It's designed by millions of years of fine tuning, and that's why year in and year out you have exactly the same populations. So we wrote a review. Couple postdocs of mine, Steve Biller and Paul Berube, and we wrote a review, and this was the cover of the review article. So we call this a federation of cells, and so this is how their artists visualized it. Why they had them marching on land is beyond me. But as I was going over, I was reading Carl's papers and his emails and everything. I found this paper by Nigel and Carl, and about biology's next revolution. And this was the figure that Nature had with that article. So I thought that was a kind of a nice parallel there. Prochlorococcus and humans marching for the biological revolution. So there they are. But what I like to think about when I look at this, which goes on forever, is if we had isolated a single Prochlorococcus and just studied that for the rest of my career, I would have been so wrong about how the oceans work that it would have been ridiculous. So we're now adding 1,000 single cells to this. So this will go and from all over the oceans, really trying to get at really what is the total pangenome of Prochlorococcus. How many different genes are there in this meta-population? And we're developing with the Joint Genome Institute a alcove for Prochlorococcus that will hold all of these data, and also will hold population data, transcriptome data, proteome data, anything, all things Prochlorococcus will go into this one place so that we can cross-reference all the data. And if there are any interested people that would like to play around with this. It's in development. Come talk to me, because I'd love to have people have some input from people. Okay. So now I'm just going to tell you a few little stories that illustrate the power of being able to study an organism across these different scales of organization, from the genome to the global ocean. And this is a story about nitrogen. Nitrogen is a key limiting factor in the oceans. If you take a bucket of seawater and you add phosphorus, not a lot happens in most places, but if you add nitrogen and phosphorus, you get a bloom. That means nitrogen is a limiting factor. And our first Prochlorococcus cultures that we isolated couldn't grow on nitrate. If you gave them nitrate as the only source of nitrogen, they wouldn't grow. They could use ammonia, but they couldn't use nitrate. Some of them could use nitrite. And that was really puzzling, because Synechococcus, its close cousin, could grow on nitrate. And it was not only puzzling, but it was disturbing, because the multiplier is huge. If nitrogen is a limiting factor in the ocean, regulates phytoplankton growth, that regulates the CO2 flux from the atmosphere to the ocean, which regulates the CO2 in the atmosphere, which regulates global temperature. This is a chain of inference that the presence or absence of the ability to use nitrate in Prochlorococcus is extremely important. We found it very puzzling, and we also really couldn't believe that there weren't any Prochlorococcus that couldn't use nitrate, even though ours couldn't use nitrate. So the hunt for nitrate using Prochlorococcus, and this is how we do our work. And this is work done by Adam Martini, former postdoc in the lab, and Paul Berube. The first thing they did was, you can go out with these pieces of DNA in the wild, and you can look for the gene that you need, nitrate reductase, to use nitrate. And then on those pieces, you can look for pieces that have that gene and pieces that also have some recognizable Prochlorococcus gene. And that was the first evidence, is, yeah, they're out there. We might not have them, but they're out there from the genomes. And so then Paul set out to then target his isolations to really capture some of these by isolating fresh seawater and only giving it nitrate, so that he could coax the nitrate users to grow, and he was able to do that. And so now we have some that can use nitrate. The interesting thing is that some of them can and some of them can't, and it doesn't follow their deeply rooted phylogeny. And we're not really sure yet why that has shaped up that way over the course of evolution, but we're starting to study how they're distributed along these gradients in the oceans to try to figure out what the selective pressures are for the ones that do use nitrate. Okay. The other interesting feature that we've begun to work on is that they're not alone out there. They live with these heterotrophic bacteria, lots of them. They're outnumbered by them, and they're, of course, feeding them, because they're photosynthesizing, and they're making the carbon that feeds. They're not the only ones, but they're important in making the carbon that feeds these bacteria. And so an interesting story emerged from that. Prochlorococcus loves to grow with those bacteria. In fact, it took us years to get them free of bacteria in cultures. All of our cultures were full of bacteria that came along with them. Here's a picture of Prochlorococcus. These are the bacteria that contaminate the cultures, is the way we used to think about it. Now we think about it as something interesting to study. Eric Zinzer and his graduate student, Jeffrey Morris, figured out at least one of the reasons they love to have the bacteria with them, and that is that Prochlorococcus, they have this tiny streamlined genome and they've gotten rid of genes like catalase that are useful for detoxifying hydrogen peroxide that is generated in photosynthesis. And these bacteria have catalase, make catalase, and it's a leaky enzyme, goes into the environment and detoxifies basically the hydrogen peroxide. So Prochlorococcus feeds this guy organic carbon, and this guy keeps it from getting stressed out, oxidative stress. So that's one of the mechanisms that we know. And it turns out that there's a whole other story that I don't have time to tell you. But I think what's unfolding is that there's an incredibly tight metabolic coevolution between Prochlorococcus and these cells that it lives with. And it has streamlined its genome because these other cells are fulfilling functions that it just said, well, I don't need to do that. And so it didn't really say that, but this is the way I think about evolution. So you can imagine the email exchanges with Carl. But anyway, but I think what's emerging is a picture of a meta-metabolism. I mean, they're all interconnected, and some of them are fulfilling functions that the other ones have let go of. And I think as more and more as we can map these interconnected metabolisms, we'll start to think about these microbial communities in a fundamentally different way. And so they developed a theory about this whole thing that is really interesting. It's called the Black Queen theory. Finally, in terms of interesting stories that Prochlorococcus has shown us is that they make these little membrane-bound vesicles that they bud off. And this is the work of Steve Biller, a postdoc in the lab. They bud off 2 to 5 vesicles per cell per generation. And that adds up to 10 to the 27th or 10 to the 28th vesicles per day globally, which is a lot of carbon that they're sending out into the ocean in these little lipid-bound vesicles. And Steve has shown they're stable for weeks. They contain DNA, they contain RNA, they contain protein and lipids, and we don't have a clue why they're doing this. But we said, well, if Prochlorococcus is making these vesicles, I wonder about other marine, and other bacteria have been shown to make these, but no marine bacteria. So Steve went out into the oceans, collected seawater, was able to separate the vesicles from the seawater, and because it contained DNA, he could sequence that DNA and say what types of bacteria were making these vesicles, and it turns out that a huge fraction of the bacteria out there are putting these little capsules of information out into the seawater for reasons that we don't understand. We have some hypotheses. I don't have time to go into it, but I'm happy to talk to people who are interested. And another mystery, and this is work done in collaboration with Wilfred Vanderdonk, who is here, who I've never met. I seem to specialize in collaborating with people I haven't met. Anyway, Wilfred discovered these genes in the Prochlorococcus, some of the early Prochlorococcus genomes that were public at the same time that one of my postdocs did, so we decided to work on them together. And these are small secondary metabolites that they make, and Wilfred's interested in them for the beautiful chemistry involved, and we want to figure out what they're doing. But the most interesting thing is, so they make these compounds, put them out into the environment, and for most bacteria that make this class of compounds, they make 1 or 2. Prochlorococcus makes about a single Prochlorococcus makes about 20, and now we've learned there's a Synechococcus that makes 60 of them. I was quite disappointed to learn that Synechococcus made more than Prochlorococcus, but I have to report it. And it's the ones that are closely related to Synechococcus that make these things. There's many of the Prochlorococcus don't make them at all. And again, we don't know what the function. In other bacteria, they're known to be antimicrobials or signaling compounds, but they're bacteria that live in slimy, close contact, and these guys live in this dilute environment. And how these could find anything that they're trying to do. We really don't know what their function is. But they have the potential collectively to produce thousands of these different molecules. We've studied the makeup, the metagenomes of Prochlorococcus out there, and there's a lot of these being encoded in the genomes of Prochlorococcus, and they're incredibly diverse. So they're up to something, and we don't know. Okay, finally, I keep saying finally. I am getting there. Prochlorococcus coexists with these viruses that infect them and inject them with their DNA, take over their machinery, and make more viruses, and then lyse the cell and spread the viruses out. And so that cell dies. And they're part of the mortality of the population. But what we've discovered is that the viruses carry genes that are very close homologs to genes in Prochlorococcus, including photosynthesis genes. So the viruses are also a way of moving genes among different Prochlorococcus cells. And so this is a lawn of Prochlorococcus, and these are plaques where a virus got there and killed all those Prochlorococcus. But the classic view of the virus-host interaction was, that the virus benefits and the host dies, plus minus. The new biology, the new image is, and this is yet to be proven, is that the viruses are just part of the system. They're a source of selective mortality that helps maintain diversity. They carry these host metabolic genes. They're a reservoir for evolution of these genes. The genes experience a different environment in the virus than they do in the host, but then they get swapped back into the host. So do they contribute to the collective fitness of this metapopulation? That's what we have begun to see them as really part of the whole. Okay, so this is Adam Martini and his students' paper where they projected what Prochlorococcus will look like a century from now. And this is the predicted % change in Prochlorococcus over the coming century. So everything in the yellows and reds means increasing. So as I say here, the future looks really bright for Prochlorococcus. Prochlorococcus likes warm water. The oceans are warming. So we're not worried about Prochlorococcus. They're going to be fine. They're going to spread their territory northward. But it might not be so great for us as the earth warms. This is from Carl. For a person who thinks they understand biology and does not know they are within, not without the system, they do not know biology at all. And that was in actually an exchange about this work about geoengineering that I've been kind of vocal about. I don't think we should fertilize the oceans to draw CO2 out of the atmosphere, because I don't think we have a clue what would happen if we did that. And so this was came from a part of an email that he responded to that. Okay, so this complexity is incredibly humbling, but what I find even more humbling is these very steady emergent properties from that complexity, the stability of Prochlorococcus out in the oceans year in and year out, and these ecotypes. And the perspective we've had in my lab is that to understand life, you need to study it across all of these scales, from the genomes all the way up to the biosphere, because all these emergent properties at each level of organization here feed back on the system and shape it as it scales up again through those levels of organization. So if you're not studying the whole, you're not going to understand it. And so I'll end with another of my favorite quotes from Carl. The future of biology lies in studying the organism and the environment as facets of a single grand problem, the nature of biological organization. And this is something that I work at simply through intuition, and I'm counting on all you physicists and theorists and to develop the theories that will underlie this. And I end with, this is a picture from one of the children's books I've written with Molly Bang. One of my favorite pictures of the Earth in all of its glory. Thank you.
So we have time for questions, so fire away.
Hi, you've talked about the pangenome and Prochlorococcus as sort of a community, and they even have friends among other bacteria. In the ocean, things move around, and these are all very dilute. So, in a couple of sentences, could you say how this coherent system is compatible with flows and the dilute nature of these things in the ocean?
In a few sentences. That's one of the big mysteries, because these things are, is Jake here? One of my students calculated the distance between, I actually had a slide, the distance between these Prochlorococcus to the next Prochlorococcus and to the next, into the bacterium and to the atom of phosphorus, and they're very far. That's one of the big mysteries. When you have a biofilm of bacteria and they're all packed together, you can kind of figure out how they might be doing what they're doing. But that that's a mind bender, and we don't, because they're influencing the milieu. There's no question. They're sucking the nutrients down, and we think, Prochlorococcus as the smallest cell is able to, well, one of the heterotrophs is smaller, SAR11, but those two guys together are setting the baseline of the nutrient concentrations, because they're not limited by diffusion to the cell because of their surface to volume ratio. So they will just suck the nutrients down to the lowest level that they get them. But we really don't. There's this sense that they're communicating somehow. They're making these compounds that they're putting out that look like compounds that would be involved in communication, but maybe they've evolved for a different purpose. I don't know. But then there are these leaky functions, like the catalase one, that play a role. So they're making these enzymes that are just leaking into the environment, but they're playing a role for Prochlorococcus. And Prochlorococcus is leaking. One of my postdocs now is working on the evolution of these metabolic pathways. And as the genome gets smaller and smaller, some of the pathways are truncated. So the products, instead of being regenerated in the cell, are just being put out into the environment. And we think that is because of a coevolution with the organism that's using it. So he's piecing together those metabolic pathways. That doesn't really answer your question, and it wasn't just a few sentences. But anyway, that's the question. Yeah. No, because the ones at the bottom of the euphotics, at the low light, they synthesize more pigment, so they can harvest more. They could be more efficient at harvesting light. The ones in the surface actually suffer from too much light, and they're very stressed. In fact, at least that's the way we interpret it. And so they make less and less pigment, but they also, this same postdoc that's been working on the metabolic pathways is working on the energy flow, that how they're getting rid of that excess energy, because there's vanishingly low nutrients, but you've got this sunlight pounding away at you, and you're photosynthesizing like crazy, but you're limited by the availability of nitrogen and phosphorus. And so some of them, he thinks, are just spewing carbon out into the atmosphere to get rid of that reducing power. But anyway, yeah, when plants are in very low light, they make more pigment so they can harvest more light.
You said that Prochlorococcus are playing a huge role in the global carbon cycle, but do you think that they are also playing major roles in the global nitrogen and phosphorus cycles, say nitrogen fixation or redox cycle?
Can you repeat the question?
Yeah.
Mostly things that are done by the bacteria.
The question was, Prochlorococcus plays an important role in the carbon cycle. Does it also play a role in the nitrogen and phosphorus cycles? It doesn't fix nitrogen, so it cannot use atmospheric nitrogen. But carbon, nitrogen, phosphorus ratios in the ocean are fairly stable, so not stable, but anyway, linked. So they do play a role in that balance. Yeah.
So I've got a follow-up question to that, actually. So the future looks bright for Prochlorococcus. As they become more abundant, are they going to be causing the global climate change to accelerate, or are they going to act as a restoring force? What's your guess on that?
So did everyone hear that?
Repeat the question.
Okay. So the future looks bright for Prochlorococcus. If they're gonna expand and become more abundant, the question is that going to help us, or is that going to make things worse? Is that going to draw more CO2 out of the atmosphere or not? And we don't know, but they're not. The image of Prochlorococcus is CO2 because they turn over so fast, that they draw in CO2, but then they get eaten really fast and respired, so that CO2 goes out. The image is that the cells that really drag the CO2 down to the deep ocean where it stays are the bigger, heavier ones, like that have shells and things and clump. But we don't think that's true, because you can find Prochlorococcus pigments in the deep ocean. So we do think they're involved in the export. Maybe we're a little biased, but we do think that they're involved in the export of carbon from the surface to the deep ocean. But I think the bottom line is that it will be worse for us, because the oceans will be warmer, they won't mix as much, there won't be as many nutrients in the surface waters. Prochlorococcus will be doing well because it's better at scavenging the nutrients than the other cells. But probably overall productivity will go down. But we really don't know. I know it's a mouthful. We wish we hadn't named it that. The question is, in the distant past, is there any evidence of warming in the distant past with Prochlorococcus? And no, there's no evidence, because there's no fossil record in a sense that, but there is in the molecular evolution, of course there is. But linking that to temperature, it would be difficult. But so no, I don't think we know much about that.
So the lipid vesicles seem to have DNA, RNA, and proteins in lipids. So are they capable of replicating themselves, or they're just inert objects?
Yeah, the question was, can the vesicles replicate themselves? And no, we don't. No, well, we don't think so. And I should have said that they don't all have that. Only some fraction. We're just trying to figure out exactly what fraction have that. In fact, there was a big uproar because for a while there, people thought all the things we've been calling phage might be vesicles. But that's okay. They're not. I mean, the phage are out there. It's going to be fine. We're nailing that down. But no, they don't have the machinery to replicate.
How do they stay their layers? So different ecotypes. Why the top layer doesn't sort of go down at the bottom? What kind of feedback makes them stay there?
Yeah, that's an interesting. So it's a balance of. First of all, the question is what makes the ecotype stay layered, and physics, of course, plays a big role. Always does. There's a little biological feedback, but so the surface is isolated from the deep water by the density gradient. So those cells in the surface are not totally isolated because sometimes it mixes, but it's a balance between how fast they grow and how much it mixes. But the deep guys don't, most of the time, except in really seasonal mixed layers, they stay down there and they're isolated. So they're pretty stable. And so there it's just a balance of growth versus death. And but so that's what but when there's deep mixing, you can see they're all homogenized. And then it will settle in, and then they'll all take their place where they belong, where they know they belong. But they don't move. They can't swim or anything, but it's just the balance of birth and death.
You described a kind of collective metabolism, sort of a loose collective. Are there variations in that? Are there places on the planet where one sees more structured interactions, sort of more sociality among the population than in other places?
The question was, does this collective metabolism differ from one place to another? And I wish I could answer that. I mean, we're just trying to get at it in one place now. But I'm sure the answer is yes, because the communities of organisms. In fact, one of my postdocs now is taking all of our cultures that we've done the genomes of, and we get the bycatch of the bacteria that have come along for the ride when we isolated the Prochlorococcus, and we just ignored the bacterial genes because we were so obsessed with Prochlorococcus. But now we're looking at, okay, who came along for the ride with different ones that were isolated from different oceans, and are there any patterns there? And so in time, I think that we'll be able to answer that question at some point, and it'll be really cool.
Wonderful talk, thank you. So you have different levels of diversity within the organism. You have your apple, so you've got your flexible component, and you have your core component. And in the core, to use your analogy, you can push a button and you can move around the core, pieces of the core. In your data, you showed a beautiful slide which showed that the genome organization of the different Prochlorococcus, they also differ. Is there a correlation? So if you throw away the flexible part and you look at the core, is there a correlation in the organization of the core genes with the ecotypes?
Whew. Is there a correlation in the organization of the core genes with the ecotypes? Yeah. Yeah. No, I mean, we haven't really looked at that in any. Those two that were compared were at the ends of the phylogenetic tree. They're like the most, almost the most different. But I don't think we haven't really looked at that. But you reminded me of another dimension of the iPhone analogy. You can spend an entire lab meeting talking about this analogy, and you can really get crazy. But it's the settings part of the gears in the iPhone. So for the core genes, for the core apps, you go into the settings, and everybody sets their core differently. So that's the analog for alleles. But the order is something, yeah, well, if you took out all of our iPhones, we'd probably find them all. Yeah. Yeah.
So a question with regards to, you were describing kind of the coevolution of the bacteria, the viruses, Prochlorococcus, and it seems very dynamic, but also it's surprising to me that it would be so stable because it seems inherently vulnerable to outsource specific functions. And so I'm wondering if you have any sense of what conditions might possibly obstruct the kind of symbiosis between them and disrupt the system.
The virus host system, or the bacterial. I don't have a good answer for that, actually. I mean, I can say read the article about the Black Queen hypothesis, because it's a very good article describing what would. It's not ours. It's Jeffrey Morris. And Rich Lenski was involved in that, but it describes the evolutionary forces that would sustain the connected metabolism and the streamlining of the genome, and it's pretty compelling.
So you've worked with a very large system. Is there a way actually that you can take now this ecosystem and do experimental evolution in control in the lab?
Yeah.
Now it needs to analyze what's going on with your
Yeah. We'd love to do that, but these guys double once every day